Probably get rolling. Okay, I'll stand over here. Great. Great. Morning, everyone. Happy last day. So we're going to finish off. This is the last kind of substantive lecture for the course. We're going to be talking about assortative mating. We've been kind of talking about assortative mating a bunch through the lecture, so hopefully a lot of these things are going to maybe fill in some of the gaps that we may have missed as we just have been alluding to assortative mating throughout all of the different lectures. I think that's it. I don't think there's any announcements. Well, we can make announcements at the final thing about dinner and stuff. Great, assortative mating. So, what is assortative mating? I think we all kind of understand it. The idea is that it's in contrast to random mating, where people base their mating decisions on nothing whatsoever, just picking random people from the population. Whereas assortative mating is the idea that people actually select who their mates are based on attributes of their mates. I think that's, the idea of assortative mating is something that's really intuitive. We think that probably this is what everyone's doing. However, an awkward thing is that in most of the stat gen methods that we've been using, we've been assuming that there's random mating, and this can have some consequences in how we interpret the results that we're producing. I also want to highlight that sorting can happen within a phenotype. So you could have like tall people who are more likely to mate with tall people, but you also could see sorting across phenotypes. So you could have tall people who are more likely to mate with neurotic people, or something like that. And both of those things will have different kind of consequences. I also want to highlight one other thing about assortative mating. Assortative mating is, you should think of it more as a descriptive statistic. I think in our heads we often think of it as, we have a causal model in our head of people making conscious decisions that are leading to the sorting that can create problems. But ultimately, it's just about correlations between spouses. And there's lots of ways these correlations can arise. Well, we'll talk about that later in the lecture. Assortative mating is widespread for a lot of different phenotypes in the population. Two really prominent ones that as social scientists we might be interested in are sorting in education and sorting in height. These are two of the strongest phenotypes where we see sorting. At least I mean, I guess there's other kinds of sorting like geography and skin color and things which are stronger than this even, I think. But these are two very prominent examples that we see sorting in the population. But there's also evidence of some sorting for other kinds of traits like body mass index, personality, political preferences, disease susceptibility. And so spouses or mate pairs often seem similar in these dimensions. Yeah.
So can you give some examples of whether they can be on disease susceptible? Like what they have done?
That is a great question. I should maybe have some examples off the top of my head, and I don't. Sorry.
Can you repeat that question?
Oh, so the question was, could I give some examples of some diseases where there's evidence of assortative mating? Yeah. So is there sorting in psychiatric disorders? I didn't know well enough.
Maybe people that know better than me. Yeah.
Well, there's sorting on health.
Yeah. So I think there's general sorting on general health, which might not be surprising to the degree that education is a major determinant of health. And so if people sort on education, then you might think that they may also be similar with respect to their health. But in terms of direct sorting, I don't know that people are assessing Alzheimer's risk when they're 20 and sorting based on I mean, I guess if you're in like some support group for if your parents both have Alzheimer's and you meet in a Alzheimer's support group, then maybe something like that could happen. But we can tell ourselves lots of stories. Great. So why should we care about assortative mating? I think there's actually two big reasons why it's really interesting. First off, assortative mating is just interesting. I think that as a social phenomenon, I think that social scientists are very interested in how people match and sort and mate in the population because it has implications for things like inequality. If wealthy people only ever marry wealthy people, then that's going to distribute wealth through society differently than if people are sorting more randomly on things like income. If people are sorting based on where they're born from, this has implications for migration. You can think that maybe is related to intergenerational mobility, and so I think that assortative mating itself is a really interesting social science phenomenon that's worth studying intrinsically. However, in our particular case as well, assortative mating has a really strong effect potentially on a lot of the types of quantities and things that we've been discussing this week. So it's going to affect things like heritability and GWAS and kind of everything we're thinking about. And so understanding how much assortative mating there are on the phenotypes that we're interested in studying is important in order to interpret those results and potentially correct those results when we're studying them. One final reason is that one consequence of assortative mating, which we're going to dive into in more detail in a few slides, is that assortative mating increases the genetic variance in the population. And so if there's greater genetic variance, that means there are more people with extreme draws of genetic risk. And if there are more people with extreme draws of genetic risk, then you would expect to see a greater prevalence of disease or other things that, if we think about kind of a liability threshold sort of model. And or alternatively, if it increases the variance, then that also is maybe related to inequality in society because there are more people with extreme draws in the education distribution, both in the left and right tail. Great. So partners may look alike for a lot of different reasons. And so this first one is kind of this causal model that I was thinking about. People may choose who they're mating with based on those individuals' characteristics. So you may just see tall people prefer to be with tall people or other things like that. And so this is one reason why you might see mate pairs being more similar. Another is social homogamy. So it might not be that people are purposefully choosing to mate with people who are similar to them, but it might be that they just find themselves in situations where people are similar to them. So for example, if you tend to meet your spouse in college, then you might see some assortative mating on education just because you're surrounded by people who are also in college and therefore who are more similar to you phenotypically and things like that. So this is the idea of social homogamy. Another reason might be it may be people are sorting directly on certain phenotypes. But the thing they're actually sorting on is a genetic correlate of the thing that you're observing. So for example, people maybe sort on intelligence. It might be that people who are brighter want to mate with other people who are brighter. And even if they don't care how much education their spouse has, it might, education and cognitive performance are highly correlated genetically. And so you might see something that looks like assortative mating, or it is assortative mating in education, even though that's not the most important relevant factor in influencing that decision. So these are all three reasons why. Oh, that is not what I have written here, but what I said was true. This one here is about disconvergence time. So this isn't about assortative mating at all, right? So the example here is like for BMI. You might imagine that if you have a couple, BMI is highly correlated between spouses in general. But that may not be because they had very similar BMI when they met. It could just be that they meet and then they live together for 30 years, and so they share the same diet and activities and lifestyles, and therefore their BMI slowly converges over time. And so this is maybe a sort of you might think of it as a confound if we were looking at phenotypic similarity between mate pairs. And it's not assortative mating, it's just mate pair similarity. Great. So through this lecture, I'm going to focus primarily on this first idea because I think that's the one that we most often think about. But it's important to realize that these things are potentially baked into the assortative mating estimates that we're going to be talking about. This was just a funny paper that I kind of liked, talking about how we might expect assortative mating to be changing over time. And so there's a paper here that looked at how the emergence of online dating affected mating patterns in South Korea, I think. Yeah, in South Korea. And so they found that as online dating became more prevalent, people sorted less on things like just geography, let's see, what is it? Less on occupation and geography, which used to be really important factors, and more on like education and other demographic traits. Which is like, I don't know, we might expect to see this. I can't. I've not done a close read of the study, so there's maybe other problems with it too. But I do think that we would expect assortative mating patterns to be changing over time as just society is changing. Okay, I want to bring up this idea here. This is a concept that David brought up in his heritability lectures, and so this is a model that's often underlying a lot of the work in assortative mating, and so it's just going back to the ideas of what leads to spousal similarity. The phenotypic assortment model is a model assuming that people are sorting specifically on the phenotype that you're interested in, and so if the correlation, so this matters for like genetic studies especially. So we might see that spouses have correlated polygenic indexes for education. And under a phenotypic assorting model, we could say, okay, well, the thing that causes that to arise is people explicitly sorting on education. The reason this is done is tractability issues. You could imagine other kinds of models that would lead to correlation between polygenic indexes. So like what other alternatives might create correlations in polygenic indexes between people, or how else might people sort on education? I think the question isn't very good. What kinds of behaviors might lead to correlations between spouses on something like education or other phenotypes? Yeah.
It could be that other outcomes that are related to education could be genetic.
Yeah, exactly. That's one of them. That's one thing. So people may be sorting on other correlated phenotypes, and if that's actually the case, then we might see different kinds of patterns between the relationships between phenotypic sorting and genetic sorting. So that's a really important one. Another one which is, I think, maybe less intuitive, you might imagine that people are actually, instead of sorting on education itself, maybe for some reason they're sorting just on the genetic or just the environmental factor. And I guess that's sort of related to this here. If people are just sorting on the environmental component of education, but for some reason they don't sort on the genetic component, maybe it's because that's just another sort of, it's a genetically correlated factor, right? So we could imagine other types of models that would lead to a correlation, a phenotypic correlation and a genetic correlation between spouses. But just to make our lives easier, we often assume this phenotypic assortment model. There's some work in EA4 where we test the phenotypic assortment model, and it doesn't look like it's actually that good either. So, but we have to do something. Great. So let's talk about how assortative mating then affects our genetic studies, because I think that's going to be super relevant for us. And so there's a bunch of this. The work on assortative mating is very old. It goes back to Fisher, 1918. There's a lot of work by Crow and Kimura on assortative mating. David always likes to point out that when Fisher was asked about all the work that he'd done specifically for this paper, which contains just a ton of everything that we still talk about today. He said everything was pretty straightforward except for assortative mating. Like assortative mating was the hard bit, and it still kind of is. It's very tricky to think about how assortative mating will be incorporated into our models. Going back to this idea, because there are lots of ways that you could try to model assortative mating. And we don't really know exactly how people are sorting. There's a lot of degrees of freedom. It's tricky. And Fisher thought so too. But there are kind of two big consequences of assortative mating on the genome that are going to have influences on our genetic studies. And so one is that assortative mating increases homozygosity at causal loci. And so the idea is, imagine there's a SNP that increases height. Then if assortative mating is happening, then a person who has this height increasing SNP will also likely have their spouse will also likely have a height increasing SNP, and therefore it's more likely that they'll pass both of those on to their children. Same if you have if you don't have it. And so you'll generally see across the genome a small increase in homozygosity at SNPs that are associated with the trait. But in general, for polygenic traits, this is very, very small. And the reason for that, right, is there's, we kind of talked about this idea the other day. If there are 1,000 loci, then there are a bunch of ways that you could find yourself at a certain genetic risk for height. Every single combination, like let's say that you have 500 of the 1,000 height increasing variants, then, there are 1,000 choose 500 ways to get to a similar height risk, and therefore, you don't necessarily see people both inheriting exactly the same things. And so as a result, the increase in homozygosity tends to be pretty little. But the factor that can be quite big is that it also induces, it's also a small effect, but it also can induce correlations between SNPs on different chromosomes throughout the genome. And this is also going to be a small effect for the same reason here. There are lots of ways that people can inherit these height-increasing alleles or education-increasing alleles. But there are a lot of pairs of alleles. If there are 1,000 height-increasing SNPs, there's probably more than 1,000. Then there are 1,000 squares pairs of SNPs that this correlation can load onto. Here's a quick illustration of actually how this correlation can arise, because this long-range LD is going to be the major factor that's causing us problems everywhere. And so this is, imagine that you have 2 alleles, A and B, and these alleles are found on different chromosomes, but they both increase height. Okay? And so let's say that you have a child who has a copy of allele A. Well, if they have a copy of allele A, then it means that they inherited that allele from one of their parents, right? Because that's where they got it. I guess unless this is de novo, but we're looking at common variants. If the parent has a copy of allele A, then parent A must be taller. If parent A is taller, then parent 2 is also more likely to be taller because of assortative mating. If parent 2 is taller, then they're more likely to have a copy of every height-increasing allele, including allele B. And if they are more likely to have a copy of allele B, then they're more likely to have passed that on to their child. And so kind of through this chain here, we can see that allele A and allele B are going to be correlated in their children if there's assortative mating. And so if there's assortative mating, we should expect that every single causal allele in the genome to be a little bit correlated. And so how does this influence genetic variance? Well, here's our model, right? We have some outcome Y, and we can decompose it into the additive genetic factor and the residual. And the additive genetic factor, we can just express this way as the sum, right? And so let's think about this. This is the additive genetic factor, and let's take the variance to calculate what the genetic variance is. And if we, the variance of this sum is the sum of the variance, like this term here, plus 2 times the covariance between every pair of SNPs. Right? So under random mating, we expect that SNPs on different chromosomes to be uncorrelated, which means that this covariance term here is going to just be 0 for every pair of SNPs that are on separate chromosomes. And there's a lot of those, right? If you chose 2 random SNPs in the genome, almost always those two SNPs are going to be on different chromosomes. And so this thing will be quite small when there's random mating. However, if there's assortative mating, then what that means is whenever beta J and beta K are positive, then this covariance will also be positive no matter where it is in the genome. And there's J times K, there's the number of SNPs squared of these terms in the genome. And so even though these covariances can be very small, once you sum over every pair, this can actually be pretty big. And so assortative mating, even though it has very small effects on things like homozygosity and LD, when we start looking at the additive genetic factor and therefore things like the heritability, it can have big effects on heritability. It can have big effects on anything that aggregates these effects over the genome. Yes?
So what happens, I'm just curious, if dissortative mating happens where taller people shorter?
Yeah, that's a good question. So we could imagine, the question's about what would happen if there's disassortative mating, meaning that spouses have negatively correlated phenotypes. Everything is just the opposite, right? And so here, parent 1 would be taller, meaning parent 2 would be shorter, and so this correlation would become negative. And so in this thing here, this is positive, right, because it has a squared term and variances are always positive. But now when beta J and beta K are positive, then this covariance would be negative, and so it actually will decrease genetic variance. I don't know that I've ever seen a really credible example of disassortative mating. Have you ever seen anything like that, Alex?
No, there's a lot of not credible examples.
Yeah.
For gender.
Okay, yeah, so sex is an example of disassortative mating, I think.
I also think like age is another example. Although age is not like phenotypic traits you are interested in, but like, typically like older men tend to marry younger women.
Sometimes. I think, so this is, I think that people bring this like, you know, colloquial knowledge sometimes to it, saying, oh, opposites attract, but when you actually go in, like, you don't, you know, I mean, again, I've not seen evidence for age, but I suspect that if, you know, I think of all of my friends, and in general, they're born within just a few years.
Of course, yeah, but on average, like, men tend to marry women 2 years younger than them.
Oh, sure, but that's not
The second marriage.
The second marriage. But that's just a mean shift, right? So the question is how if men marry women who are a little bit younger than them, but men who are a little bit above the mean among men tend to marry women who are a little above the mean for women, then that's still sorting. Mm-hmm. So it's so yeah.
I
So
think there might be examples from non-human organisms,
okay.
but I'm not sure there's any convincing ones in humans.
Yeah.
You know, immune-related genes.
Mm-hmm. Yeah. So Alex is saying perhaps there are non-human examples that exist, but yeah, I've never seen a human example that was very credible. But theoretically, it could exist, and if it did, it would actually decrease genetic variance. Another thing about assortative mating is that it, I said it increases genetic variance, but it does so over time. It doesn't go from one generation to the next increasing genetic variance. It will increase, pretty quickly, but it'll eventually approach an equilibrium. So it'll asymptote to some higher value if you go from a random mating state to a sorting state. So let's talk about exactly why that is. So I'm going to code these X stars as genotypes, but I'm going to just say that it's a single, it's looking at a single chromosome, a SNP on a single chromosome, rather than the 0, 1, 2. So now these are 0, 1, right? So if we wanted to say what's the covariance between SNP J and SNP K at time equals 0, well, there is a 50% chance that these two alleles were inherited from different parents, and there's a 50% chance that they were inherited from the same parent. Right? So I'm gonna split this covariance into two pieces like this, and I said it was a 50% chance, so I'm gonna put the 50% there. Now, something to notice here is that this term here is the covariance between the alleles inherited from different parents, and so this is sort of a measure of how much sorting there is, at least how much genetic sorting there is across these two SNPs in the population. This one here is the covariance within a single person 1 generation ago. And so we can say that this covariance here is just the covariance at t minus 1. And so this relates to how much genetic, and since these covariance terms are related to how much the genetic variance has expanded, we can see that this defines the evolution over time. And so every generation, we're going to move halfway between how much sorting there was in the current generation and then how much sorting we should expect to find due to the amount of sorting in the population. So we just kind of move halfway every generation. I think that this, Alex showed me some results suggesting that this is slower if there are things like indirect effects. Is that right?
Yeah. I mean, that whole thing gets even more complicated.
Everything gets more complicated when you have indirect effects interacting with assortative mating. Yeah. But this general principle of when there are changes in sorting, that the effect on the population, the genomes of the population in terms of additive genetic variance and genetic correlations and things like that will move towards the new equilibrium over several generations. And so that's important too, because we often talk about equilibrium heritability versus heritability under random mating, and people will talk about these two different concepts. And that's what they're referring to here, is like, what would the heritability be if people were randomly mating versus, what would the heritability be if people continued sorting at this level, over infinite generations. Oh, and this is the point that I just made. This corresponds to how much sorting there is in the parents, and this is the covariance in the parents' generation. Okay, so assortative mating biases everything. All of our genetic studies that we've been talking about. So for example, if you're doing a twin study and there's assortative mating, then you would see that DZ twins would have a correlation that's greater than 50%. And that's, in twin studies, we generally assume that at least the simple twin studies, we assume that the correlation of the additive genetic factor in twins is 0.5. And so if we don't account for assortative mating in our twin studies, we're going to get biased answers. And we actually, David had a slide about how you might incorporate assortative mating into those studies to correct for that. But if you don't take this into account, you'll get biased estimates in your twin studies. In GWAS, so remember when we said GWAS summary statistics, they not only correspond to the effect of the specific SNP that you're interested in, but it also corresponds to the effect of every SNP that's in LD with that SNP. Assortative mating induces LD between that SNP and every other causal SNP in the genome. And so if there's assortative mating, our GWAS estimates are going to be inflated as a result. Alex talked a little bit about this in his lecture too. Assortative mating can also inflate our heritability and genetic correlation estimates. And so think about like LD score regression. LD score regression is based on GWAS summary statistics. If all of our GWAS summary statistics are inflated due to assortative mating, that's going to inflate our heritability estimates from LD score regression too. So I think that's pretty intuitive. This is the really interesting result that that Alex also mentioned. It's super interesting, very curious. If you try to estimate heritability with Greml, and you do it in an infinite sample, it turns out the Greml will spit out what the heritability would be if the population were randomly mating. A bit funny. So even if your population is sorting a lot and you have an infinite sample, it's going to give you the random mating heritability. However, in a finite sample, it's going to be biased towards the random mating. Is that right? Isn't it always in between the two?
Yeah, that's what they claim based on simulations, at least.
Okay.
It's between the equilibrium and the random mating.
In general, it's upward biased, but the amount of bias depends on the sample size that you use to estimate it.
With bias, depending on which parameter you think is the one you should be estimating.
I guess that's true. Yeah, that's true. I guess it's biased away from its asymptotic value. But yeah, this is kind of curious behavior, but assortative mating messes this up, is the point. Yep.
We typically calculate LD scores within a centimorgan of the target SNP, and if there's long range LD from assortative mating, would the LD score intercept be too small if it's not capturing cross chromosome, like true cross chromosome?
Yeah. So the question is how assortative mating would affect the LD score intercept. So I think that, so if people are sorting on the phenotype that we're studying, then I think all of the beta coefficients are inflated by a proportionate amount. I haven't proven this, but this is the intuition that I've that I have. And so if we just imagine we've multiplied all of the betas by 2, then there's going to be a relationship still between the GWAS betas and the LD scores. And so that's so that bias then gets wrapped into because it's correlated with the LD scores, it gets pulled into the heritability side and wouldn't really live in the intercept. To the degree that people are sorting on other things, or that the inflation is not just inflation by a constant, then yeah, it'll live over there in the intercept. In practice, I think that even when there's sorting, it can look like there's not stratification and that heritability is inflated. So I think it's the case that you just can't detect biases from assortative mating in general, at least phenotypic assortative mating.
And also, wouldn't it throw off the LD scores too if you're not looking across chromosomes and assortative mating is inducing LD across chromosomes?
Yeah, I mean, in general, yes. So the question is, wouldn't it throw off the LD scores? But if the LD score is proportionate to kind of the amount of LD that's existing on other chromosomes, right, then it's those things are correlated, and so it's going to pick up the omitted signal, even though your LD score is based on a narrow slice. I haven't done this super carefully, and so it may be that my intuition is wrong.
Yeah, that makes sense.
But yeah, it's ultimately going to matter depending on how much the LD scores are correlated with the LD that you've omitted.
Right. Why might you expect the amount of off-chromosome LD to be proportional to the local LD that's not assorted?
Yeah, that's a great question. As I was saying it out loud, I got nervous.
That would be true under a polygenic model, wouldn't it? Because the more LD you have locally, the more genetic variation you're tagging. The more genetic variation you're tagging, the more LD you have with the rest of the genetic component of the trait.
That's right, yeah. So if you're tagging a lot of the genetic signal, then it's going to increase the covariance just locally even more. Yeah, because the amount that the correlation between pairs of SNPs increases is proportionate to how, it's not proportionate to the true effect. I think it's proportionate to the the marginal association of the SNP with the phenotype. I think that's true. Okay. Good. Okay, so here's also, we talked about Mendelian randomization yesterday. Assortative mating will cause problems for Mendelian randomization too, potentially, right? Because one of the key assumptions of Mendelian randomization is the exclusion restriction. And for our studies, what that implies is that the SNP can't be an LD with a SNP that violates exclusion, right? And so, we might think like, oh, well, let's just carefully choose our SNPs to ones that have known biological mechanisms, and then maybe we're okay because linkage is local, and so, we at least know that this association that we're using in our Mendelian randomization, corresponds to the genes that are in this area, and we know what these genes do. Like, we might feel safe doing that. However, assortative mating makes every causal SNP associated with each other, and so now, if you're doing Mendelian randomization for a phenotype where there's been sorting, your exclusion will be violated if any SNP, any causal SNP in the genome violates exclusion. And so, assortative mating can cause problems here. It means that the exclusion restriction in our MR is a much stronger assumption than it would be if they were random mating. And this is the point we're making before. Assortative mating and indirect parental effects interact in really complicated, messy ways. I think that just your intuition should be that it exacerbates these kinds of biases that you're seeing. So all of the biases that we talked about with indirect parental effects, you should just think they're worse if there's also assortative mating in the sample. Yep.
So it just occurred to me that like the two model phenotypes that we've been talking about a lot this whole time, height and educational attainment, are two that have strong amounts of assortative mating. So are there any like GWASs of things or any phenotypes that are especially unbiased by assortative mating that we can, look at how results are systematically different in this realm?
Bone mineral density?
Wow.
I think that's one that the Fisher Lab always likes to use.
So you don't look at someone's deal.
Yeah, you don't. You don't submit your bone scans to OkCupid, usually. Although the type of person who submits their bone scans to OkCupid, maybe people sort on that. I say the same thing about like these dating sites that are rising, where you submit your genotypes and they try to match you based on like your genetic, propensity. I don't know that those. I'm pretty confident that actually those are totally meaningless, except that the type of person who signs up for such a dating company, they might be good matches.
They didn't pay me to say that, but there's actually a Netflix series on that.
Oh, okay, yeah. Netflix. There's a Netflix series on everything these days, huh? Great. Okay. We can all watch it. Great. I also want to do talk quickly about assortative mating versus inbreeding. So inbreeding is when people who are closely related have offspring. And so generally, it's not attached to some phenotype like assortative mating is. So assortative mating, we say people are sorting on education or things. I guess if you wanted to say that inbreeding is assortative mating on like who your parents are or something like that, then I guess it's assortative mating in that sense. But they look different genetically, in that if you have inbreeding, this leads to long runs of homozygosity, meaning you'll have just big strings of SNPs where you're homozygous at every single site in the string. And you can imagine why that is, right? If you have siblings who have offspring, then they may both pass on their paternal chromosome to their child, and the paternal chromosome is going to match exactly at every site, including incredibly rare variants. And this is why when we have inbreeding, it means that there's higher chances of dominance effects for rare variants to become very common, because if one of your parents has a very rare variant that has a large dominance effect, it's unlikely that you would inherit that rare variant from your other parent. But if there's inbreeding and you've just inherited the same chromosome, then you're going to get 2 copies of that rare variant, and so that can have potentially big impacts on their offspring. Okay, so now I'm going to talk about a couple methods that have been developed that were designed to test for or estimate assortative mating in populations. Let's talk about how we might do that, right? So if we just had whatever data we wanted, so we called on the data gods again, and we said, I really want to study assortative mating, what can you give me? Ideally, the thing maybe we want to do is just estimate it directly. So we'd say, give me the phenotypes of a bunch of mate pairs, and I'll just take a correlation of all of them, and that might give me a measure of how much sorting there is for that phenotype in the population. And so we, if we're looking at the correlation of phenotypes, and we often refer to that as phenotypic assortative mating. Alternatively, maybe instead of looking at the phenotype correlation, maybe we want to look at the correlation of genetic risk or polygenic indexes. And so we could also ask the data gods for the polygenic indexes of all of the mates in the population, and look at the correlation between them. And we often refer to that as genetic assortative mating. So why would these two things be different? Why might we prefer one of these to the other one? Do they give us the same answer? Why not?
I think phenotypic correlation could be from many different reasons, like environmental.
Yeah, no, that's exactly right. Yeah, so it goes back to this idea that we had before. So let's say that we were looking at sorting on BMI, right? And so there is generally a strong phenotypic sorting on BMI in the population, but just looking at a mate pair's BMI at a certain moment in time, especially if they've been together for a very long time, we can't know whether or not they were very similar when they first met, or if they're similar now because they faced really very similar environments for a very long time. And so that's a reason why maybe looking at something like genetic assortative mating might be more interesting, because a person's polygenic index was fixed at conception and therefore fixed when they met, and so they can't influence each other's polygenic index. Or there aren't common environmental things that can affect their polygenic indexes either. And so there's been some focus on this idea of genetic assortative mating as well for that reason. There are other technical reasons why this is a bit tricky, because we're not actually, in general, if you want to study assortative mating as a social scientist, you're actually interested in the phenotypic assortative mating, right? And so going from genetic assortative mating to phenotypic assortative mating requires additional assumptions. So we might assume the phenotypic assortment model. But if we don't believe that model and what we actually care about is phenotypic sorting, maybe other designs are better. If we had maybe longitudinal data from when a person was young perhaps that could, since we have the data, God's giving us our data, maybe that would be better. So another thing that we could do, so let's say that, the problem with all of these kinds of studies are that they require data on mate pairs, and data like that are difficult to come across potentially, right? And so maybe there's a better way to do it if we have population-based data on unrelated individuals. And so, one thing we might do is ask people, how much schooling did your parents get? We could do something like that, and then just use the correlation based on the reports of their children. So the benefit of that is we could potentially do that in a very large sample. What's the problem with that? Yep.
Measurement error?
Yeah. There's probably some sort of error in, a person may not know when their parent dropped out of school or not. Or they may, and the error may be correlated in ways that affect your estimate. So that's the problem with that. There are methods that have been developed, though, to infer how much assortative mating must have existed just by looking at the genes within an individual. So we're going to go through two methods. Three, I guess. I'm not going to talk much about this one. This is one of the first papers that did this, where they looked at individuals. Oh, actually, no, this is not. This is an example of spouse pairs. Sorry. So in this paper, I think they identified the spouse pairs in the UK Biobank by matching them on a dress and things like that. And they measured how similar the phenotypic correlation was between the individuals in the UK Biobank versus some measure of genetic correlation between the spouses. And I'm not going to spend too much time on this, but for height, they see high phenotype correlation, and these are the measures of correlations based on genes, and we see that those are also quite high. For BMI, though, they see high phenotypic correlation, but reduced correlation when they use their genetic measure. So this goes back to this idea that we've talked about a couple times, where the phenotypic correlation and the genetic correlation may tell us different stories. So this is an example of a paper, though, that infers whether or not there was sorting on some phenotype in the population by just using samples of unrelated individuals. And so let's talk about how they do it. So imagine that you have 3 individuals, and the green dots here for each of these rows correspond to alleles that increase the trait that we're interested in. So if we're thinking about height, then each green allele increases height, and each blue one is associated with decreased height. And so this person is predicted to be the tallest, and then the medium, and this person is predicted to be the shortest of these 3. And if there were, now let's say that we built a polygenic index for this individual just based on their odd chromosomes. Well, if there's random mating, then we wouldn't expect any correlation between the SNPs that a person inherited on odd chromosomes relative to the ones that were inherited on the even chromosomes. And so what that means is we created a PGI for height based on the even chromosomes, then we wouldn't expect to see any correlation between predicted height for the odd chromosome PGI and predicted height for the even chromosome PGI. However, if people are sorting, then you would expect to see a correlation between these two odd-even PGIs. And so over here we see this is predicted to be the tallest based on the even chromosomes. This person's predicted to be the shortest based on the even chromosomes. And so this would be evidence of assortative mating for height. And so that's the premise of this idea. So they're just going to do a comparison within an individual of the alleles that they've inherited based on these principles of how assortative mating affects long-range LD in the genome. I think this is just saying in words what I just said out loud. Yeah, this is just saying everything that I just said. So there's kind of 2 potential confounds to this analysis. So one is if there's sample overlap. So if you built your polygenic index in the same sample that you're using to estimate assortative mating, then the error on the even and odd chromosomes will be correlated. And so you might see correlation between these 2 PGIs, even though there's no assortative mating. And so that means that you need to estimate your GWAS in a different sample than you're estimating assortative mating. I think it's unsurprising. The other one is population structure. Population structure is a bit funny because you can think of it as also a type of assortative mating, right? So if let's say in the UK, all the tall people live in the north, and all the short people live in the south, is that right, or is it opposite? Where do tall people live in the UK?
I think either is true. But I think it's actually taller, and the average height is taller in the south.
In the south. Okay, so taller people in the south, in the UK, right? And so if we measured, and then people tend to find their mate just based on where they live. And so, we might see that if we were to look at the correlation of mate pairs in the UK, that it looks like people who are genetically predicted to be taller are mating with other people who are genetically predicted to be taller. But it's not because there's any sort of social preference for tallness or shortness. It's just because people in the south are having children with people in the south. And those people are taller. And so, on one hand, even if the assortative mating is being driven by population structure, like, that is assortative mating. But if we want to get a measure of assortative mating that's more interesting from like a social sciencey point of view, what we might do is residualize our PCs first, residualize our PGIs using PCs, and hopefully that residualizes any sort of correlation out that's due to population structure. And we have something that more closely resembles kind of direct sorting on the phenotype. So that's a thing that we could do if we want to have something that's maybe a bit more interesting, a bit more interpretable. So what does Loic find? So he did a bunch of work, and he can actually calculate how much he would expect the correlation to be between the odd and even chromosomes based on the phenotypic sorting that's happening. And so for height, in these 4 different data sets, he estimates how much sorting there is, and he sees pretty strong correlation between the odd and even chromosomes, and so there must be pretty strong sorting for height. Same for educational attainment. There's pretty strong sorting, or pretty strong correlation between the odd and even chromosomes, and so that must mean that there has been sorting on education. Here's the example. So they tested on bone mineral density. I guess there's some negative sorting in JERA. But I don't know if that survives Bonferroni or not. But in general, you see that there's really not much going on here. And so this is sort of a negative control. You don't expect to see sorting on bone mineral density, and it looks like there's not. Great. Got 20 minutes. Okay, so this is some work that I've worked on, where these are sort of tests of assortative mating. We can look in the genome and see, has there been assortative mating any time in the past? We may be interested in the actual level of assortative mating and how that's been changing over time. And so Miles Kimball and I developed a method, Grant also helped out a lot on this project actually, that allows us to look at the long-range LD in the genome, at SNPs that are associated with different outcomes, and infer how much sorting, what the parental correlation must have been going back in time, at different points in time. Instead of splitting it, looking just at odd even chromosomes, we look at every SNP pair, like every cross-chromosome SNP pair in the genome. But other than that, it's the same principle. We're looking at how much cross-chromosome correlation there is, and using that to infer how much sorting there is in the population. So here's a bunch of examples that I think illustrate some of the things that you might learn in a study of assortative mating. So first off, I wanted to say, where a person's from is assortative mating. And so we could, in theory, create a polygenic index for latitude of birth within the UK Biobank, and estimate how correlated predicted latitude of birth is between pairs of individuals in the UK. And so if we do that, and we don't do any kind of stratification correction, so these are just the raw PGIs, we see that at the beginning, so these are individuals, so parents who are having children, at about 1940, there's a moderate level of correlation, but it's quite imprecisely estimated. And then we see that it, possibly decreases, and then during like World War II, maybe, we think this is maybe a World War II effect, but then after the war, when people are kind of returning back home, there's kind of high levels of sorting again between predicted latitude of birth for these individuals. So I think this is a noisy but kind of cute example of how assortative mating might tell us something about migration. Just to make you feel good about this, I told you that if you residualize the PGIs for the principal components, that it removes stratification. Well, this is 100% stratification, right? And so if you residualize the polygenic indexes for the PCs and estimate sorting at that point, then you just get zeros across the board. So this is just sort of another kind of comforting negative control. We would expect to see zeros, and we see zeros. So we've been talking a lot about height, because height's an important example of sorting in the population. And so when we use the height PGI and estimate sorting over time, we see that, as has been shown in a bunch of studies, sorting on height is quite large, and it looks like sorting on height is increasing over this interval where we have data in this study. So that's potentially interesting. Yeah?
So the predictability of PGI for height changed over time.
So, do you know, I think I've unlinked these. In this data, it doesn't look that way, actually. I think that it's pretty flat. If you look at the R-squared of PGIs for people in different decades over this time period, the R-squared is about the same every year. But it is true. If you could worry that so this is an important point. If the PGI was much more predictive in this cohort for height than in this cohort, even if people were sorting the same amount, so the correlation of phenotypic height were the same over time, what you would see is that the correlation of the PGI for height would be increasing. So that could explain this increase in slope. But when we go through and we actually test, has the predictive power of the height PGI changed in this sample? We don't see that. It's pretty stable. That's height. So let's think about educational attainment. So lots of people have looked at sorting on education. I think this is an important social science question of how much are people sorting on SES-related measures. There was a paper that came out in 2014 that said, sorting on education has been increasing. But then there were a few follow-up papers that said, no, the increasing sorting that was reported in this paper is just a technical artifact due to the way that they coded the education variable. And so. And this goes back to this issue of if we're looking at phenotypic assortative mating, it can be sensitive to how you're coding, how you're reporting, things like that. When we use the genetic evidence to see how much sorting, we see a pattern that's kind of similar to the education, where there is sorting in education, and that the sorting has been increasing over time. And then going back to your point, if we look at the R-squareds, it actually the predictive power for these cohorts is a little bit smaller than over here. We did not separate women and men in this sample.
Because, Pamela Health, their paper in ASL basically argued that the predictability of PGI for educational attainment increased mostly for women, because in the past, women have some structural barrier to the higher education.
Mm-hmm.
But, after 1960 or 1970 cohorts, they are moving to the higher education more and more.
Yeah, I mean, it's not totally clear that structural barriers would increase or decrease the R-squared, though, right? Like you have to think. So the structural barriers would maybe put a cap on how far they could go. But if you're wondering how much variation is explained in whatever variation is left after accounting for the structural barrier, So, but at least when you don't split by sex and you just look at the raw R-squareds in the data that we use to do this, it's I think you actually see a slight decrease, if anything, over time. So we've been talking about BMI as this example. The whole time there's a high phenotypic correlation of BMI. So is this due to actual sorting or a common environment? And when we estimate assortative mating for BMI, we see this is positive. But I've tried to maintain the scale in all of these, and so the sorting on BMI seems to be much smaller than the sorting on these other phenotypes. So. So it seems like the correlation of phenotypically is more common environment than actual sorting on kind of predicted BMI due to the polygenic index. Great. So let's think about autism. And so there's a lot of autism research, and some people have suggested there's been a big increase in autism rates in the population in the past decades. Some people have suggested that a reason for that might be because of assortative mating, and that autism is just an extreme draw on kind of high performance, things that are associated with high education. And because people are sorting more on education, this will increase the genetic risk for education, but also genetically correlated traits like autism. And therefore, this increased sorting on education will lead to extreme draws on the autism risk spectrum. And so it might just be that there's more people being pushed out to the tails of autism risk. And so, so if that's the case, then what we could do is we could look, well, is there sorting on the autism PGI? When we try to do this, we don't see much. A caveat here is that the autism PGI is not very predictive yet. And so I suspect that that's driving down this estimate somewhat. And so it may just be that we don't have enough data to detect the sorting that exists for the current PGI, or it may be that as we increase the sample size for the autism PGI that we might see something here. So that's actually a caveat for all of these things. The correlation between the relationship between the sorting on the PGI and the sorting on the phenotype's a function of the predictive power of the phenotype as well. And so that's a thing that you need to take into account. But going from PGI sorting to phenotype sorting requires assumption about the assortative mating model. So here's the last example. And so we've been talking about within trait assortative mating this whole time. But I said at the very beginning that you can also see sorting across traits, and that's going to affect our estimates. And so here's an example. If we look at those plots, the heat maps that Elliot was showing us yesterday about the genetic correlation between lots of things, we see that education is genetically correlated with a ton of phenotypes, especially psychiatric phenotypes. And so, for example, the genetic correlation between educated people, between education and depressive symptoms is negative 0.3. So highly educated people tend to also be very sad. Or happy? Wait. Depression, education goes up, depression goes down. They're happy. I wonder if I coded that wrong, because I remember it being the other way. In any case, education does something to us. Or at least it's associated with something. That's a strong statement. Education's associated with something. Okay. The question, though, is, this RG could be driven by long-range LD, or these estimates could be driven by long-range LD from assortative mating. And so we can if we can difference out all of the RG that's due to long-range LD, or all of the RG that's due to long-range LD, we can maybe get an estimate of how much of this genetic correlation is being driven by assortative mating. If we do that, and again, we're looking over time, we see that some fraction of the RG that we're estimating here is actually just being driven by assortative mating in the population, and that perhaps we're overestimating it by maybe a quarter if we're going to average over this whole data set. And so when we're interpreting our genetic correlation estimates, we need to keep in mind that they may partially be driven by just assortative mating.
So can you explain more or less how that is done?
Yeah. So so you can imagine making a polygenic index for education and a polygenic index for depressive symptoms. And if you took the covariance between those polygenic indexes, let me just write this down. If I can pick up these paper towels. So imagine that we wanted to take the covariance between the additive genetic factor for education and the, well, I guess PGI, we'll say. PGI for education and the PGI for depressive symptoms, right? Well, that's going to be equal to the covariance. I'm going to rewrite this as the sum of, XB education, XB depression. Right? And so it's the covariance of these two sums. Now, some of these, so we could rewrite this as the pairwise sum of a bunch of little covariances, SNP by SNP. And some of these pairs in these little covariances, so I guess I can write this down too, right? So we're going to sum across J, sum across K, BE, BD, covariance x, j, x, k. Okay? Now, some of these covariances are within a chromosome, and so those are likely due to, these may be covariances due to linkage or, non-assorted mating thing. But a big chunk of these things are covariances due to cross-chromosome LD. And so, what we do is we just have a way of subtracting all of these out. We do that by making chromosome-specific. So you could imagine looking at the correlation between a polygenic index just based on chromosome 1, and just based on chromosome 2, and just based on chromosome 3. And so that will give you the covariance that's just due to within chromosome pairs. And if you compare those covariances to the covariance using whole genome polygenic indexes, then that kind of gets you towards this thing here. But that's the basic premise. We just try to difference out how much of the variation, or covariation in this case, is due to cross-chromosome LD. Yep?
What do you think, and you might not have an answer to this, but what do you think is causing this over time, this kind of increase in assorted mating? Do you think it has to do with, like you were saying earlier, this kind of reaching a new equilibrium with heritability over generations? Or do you think this is due to social factors or both?
Yeah, I don't know. I'm not an expert on the actual kind of social science underneath this. I mean, I think it's not surprising that we're seeing people. So I think that like this pattern here is probably largely driven by the changes in sorting in education, for example. Because I think we do see that people are more likely to marry later, and therefore they're probably meeting their mates once they're in college or grad school or late in their career or things like that. And so there's a lot of social sorting that happens just due to people staying in school longer. Whereas, again, this is just based on I don't know the data, but my sense is that 50 years ago you married the person down the street, whereas today maybe you've moved someplace to based on your professional or educational activities, and you meet the person there. So it could be lots of different things, but great. So just a quick summary slide. Assortative mating can create or exacerbate all the, so we've talked about all of these different problems, and assortative mating makes everything worse in terms of the biases and things like that. So it's good to know whether it's there and how it might be influencing the studies that you're doing. However, if you control, as Alex told us yesterday, if you control for parental genotypes in your study, that completely accounts for these sorts of biases. And so this is the kind of first best way to account for assorted mating in your studies is to do family-based things. It's not always possible, however, but there are ways to estimate how much assorted mating there might be in your sample using samples of unrelated individuals, using either like the method I showed you that Loic developed or the one that I just showed you that I'm working on. And so it's an interesting and important and difficult concept that we should be aware probably has big impacts on the outcomes that we're studying as social scientists. So that's it. Are there other questions? Oh, yes.
Sorry.
No, I'm glad.
So when you talked about the phenotypic and genotypic assortative mating, can you talk a bit about like how phenotypic or genotypic correlations might matter for friendship networks?
For friendship networks?
Like how you choose friends. Is there evidence of like genotypic correlation there?
Yeah, so I mean, Dalton Conley and co-authors has a paper where they looked at genetic similarity in friendship networks. I think that in the paper that actually came out, I think they made much stronger statements than like since then people have re-looked at their data and said that this is seems to be more representative of social homogamy than actual sorting, and so I think there is work, trying to look at how similar people are sorting genetically. Although I think that the credible estimates that have been generated that I've seen suggest that it's smallish, but still may be present. But I think it's very interesting. It's if you have the right data set and strong enough genetic predictors and adequate ways to control for social homogamy and population stratification, then I think that'd be very interesting. Yes?
So does the genetic assorted mating analysis, does it limit the sample to those with children, or it covers all couples regardless of whether they have children? Because assorted mating could be problematic for genetic behavioral genetics because it biases the heritability and other sorts of the reproduction. That's
Yeah. So the question was, in these studies of assortative mating, do they restrict to individuals who just have children? And you'd think that, that obviously would be important to consider, right? Because if people are sorting but they don't have, if they're sorting and they don't have children, then your population just dies. But you would think that how many children different individuals have will have different effects on the population. And in the case of these studies that are based on unrelated individuals and inferring how much sorting there must have been in the previous generation, right? I guess effectively yes, because we're studying the people's children in order to infer how much sorting there is in the parents. I think it's been a while since I looked at this, but there's another paper which I think is on the reading list where they measure phenotypic assortative mating. And I think that they did try to weight their estimates by how many children these couples had. It seems like that would be the right thing to do especially if you were interested in the effect of assortative mating on, the genes of the population. If you're just interested in how people are sorting, if you wanted to look at assortative mating in LGBT individuals, then just because you're interested in how similar are these pairs that are forming, then maybe the number of kids wouldn't be very interesting. And if you're interested in assortative mating among people who aren't having children, then you really can't do that genetically because there's not children to estimate it with. So that's a great question. That's another way in which phenotypic and genotypic assortative mating are different, is that it's intrinsically weighted by how many children these individuals have. That's a great point.
You can still look at the correlation of the PGI.
That's true. Yeah. So if you didn't want to do phenotypic assortative mating, but you wanted to have all the benefits of genetic assortative mating with respect to it being fixed at birth, you could look at pairs of individuals and just look at the correlation of their PGIs. So maybe you get the best of both worlds, but limited sample theoretically. I guess not theoretically, practically. Yes?
Just out of curiosity, has there been much work done on assortative mating on political orientation and religious beliefs?
I'm sure that there has.
May I?
Yep.
I think this is actually the biggest predictor of assortative mating.
Yep. Yeah.
It's
So
more than height.
maybe in the US.
Yeah.
And in Japan, we don't know the political attitude.
It's Australian data. Yeah.
Oh, it's Australian data.
Yeah. So I'm sure there is. I don't know it.
It's funny that some of the seminal studies on this were done in your home country.
Really?
Yeah.
That's interesting to me because we don't.
If you want to do genetic assortative mating on, I always forget there are aspects of political and religious leanings that are highly heritable. And so if you wanted to look at genetic assortative mating on traits that are not very heritable, it becomes difficult. Which is it? It's like participation that's heritable, but left or right that's not. I forget. What are which are? Do you?
Well, when it comes to religion, which religion you follow is not heritable at all, right?
Mm-hmm. But how
religious you are
yeah.
is heritable.
Yeah. So if you wanted to say what's the genetic sorting on if if you're Hindu or something like that, then that might be difficult to do with a genetic design. Was there a question? I thought I maybe saw a hand over here.
Oh, I was just going to say another thing that I think is kind of interesting about political and religious assortative mating is that there's surprisingly little amount of assortative mating in personality traits, and most of it's driven by political and religious values. So you really see it with mostly openness to experience, but then not the other big 5 traits. Just kind of curious.
Great, well thanks everyone. So I think that we're gonna have a little break now, and after the break then we'll start student presentations, is that right?
Yeah, and 11.
Great, see you at 11.